Hello beautiful people, welcome back to my channel once again with another new vlog. So today we are going to start our new vlog with a new topic that is a very basic topic that all of you all know. I think that's the order of draw or blood collection. So here we are, as you all know that we have started our preparation series for all the prometric aspirants and also for the individuals with their academics who are studying their graduates and diplomats in lab technologist or technician. So this is the very basic topic. I know that everybody who are medical lab technologists, they know regarding the order of blood and blood collection. So I just want to share you something in detail because as I have received this question in my prometric regarding the order of draw, and they are asking the sequence of order of draw. So I would like to share this video in detail with you all before starting our preparation series with microbiology. I just want to add up this topic in your series. So let's get started with our today's vlog regarding the order of blood draw. Before that, I would like to request you to please like, share and subscribe to my channel. Keep supporting me, and let's get started with our today's video. So friends, what is order of a blood draw? So blood collection tubes must be drawn in a specific order to avoid cross contamination of additives between the tubes. Okay, so the recommended order of draw for blood collection tube is, so here we are having a sequence of blood draw, that is blood collection. So why we are following it, because to avoid any cross contamination with that of additives is nothing but the chemicals which are present in the vacuolators of tubes of collection. Okay, so the recommended order of draw for blood collection tube is, so our first tube for order of blood draw should be blood culture bottles or tubes. As you all know, blood culture should be done without any kind of contamination, so it should be in a first order, should be from blood culture bottles. And the second is for coagulation tube, that is light blue tube, coagulation tube, light blue top, which we are using for PT and APTT purpose. So the third order of draw should be in a non-additive tube, that is red top, red vacuolator, which is it is not containing any type of additive. Okay, and the fourth is SST, that is serum separation tube, contains a gel separator. After centrifuging, you will get serum separated. Okay, and you're having a gel in their bottom. Those tubes are called as SST, that is serum separation tubes. And the next is fifth, that is sodium heparin, that is dark green top, sodium heparin. And the sixth one is EDTA, it is of lavender or purple top, and with that we can add ESR tube, which will be with a black top. Some will be collected in vacuolators, and some labs they will collect in a Wintrop's glass tube. Okay, and the seventh one is sodium fluoride tube, that is gray tube, which we will usually use to collect the sugar samples. Okay, so these are the sequence of different types of vacuolators we are using in the blood draw. So what one important thing you have to be remembered, that all the tubes or vacuolators must be mixed thoroughly as to avoid the erroneous test result. So here we are having a this is in this also we have a kind of mixing, right? So we can use blood mixer machines, or if not, if you are mixing with your hand, then you can mix it. For different tubes we are having at different times, we have to mix like 3 to 5 times, 8 to 10 times. Okay friends, so in detail about the mixing also we will be discussing in this video. So first of all, this is the correct sequence order for blood draw. So next, now you will be having a lot of questions like EDTA and everything tubes. Why we are collecting a sample, CBC sample in EDTA? What do you mean by EDTA? EDTA is nothing but ethylene diamine tetraacetic acid. So what is that? It is a kind of additive which will not get clotted. Okay, it is an anticoagulant, that is against coagulation. It will not clot the blood. So it is a chemical that binds certain metals ions such as calcium, magnesium, lead and iron. It is used to prevent blood sample from clotting and also keep the bacteria from forming a biofilm. Biofilm is nothing but a thin layer stuck to a surface. So this is regarding your EDTA sample. Why we are collecting in a EDTA? EDTA is example is full form is ethylene diamine tetraacetic acid. Why we are collecting the samples in EDTA? Because we don't want the samples to get clotted and we don't want any kind of biofilm in our sample. So this is the reason why we are collecting in EDTA. Now the second is in sodium fluoride. Okay, it has been known to have an anti-glycolytic effect that inhibit glycolysis by erythrocytes. So sodium fluoride, normally we are using this tube for glucose testing. So why we are collecting the sugar samples in sodium fluoride means in the sodium fluoride there they will stop the glycolysis process by erythrocytes. That's why we are collecting the sugar samples in sodium fluoride. The next is note. Now comparing plasma and serum SST results of glucose values have an average difference in their values. So there is no difference in glucose values collected from plasma, NFA tubes or serum SST tube. So friends, nowadays as you can see we are collecting the sugar sample in gray tube. If not, we can also collect the sample in a SST tube, that is gel tube, because as per the scientific research, they have been known that there is not much difference between collecting in gray tube and or gel tube. So you will be having an accuracy in between both the results. So now if you don't have a gray tube, no need to worry, you can collect in a yellow gel tube also. Both can give you an average accuracy values. Okay, now that is sodium citrate. Sodium citrate, it has been used in a coagulation test because of the factor 5 and factor 8 are more stable in citrate specimen, and EDTA used for hematology test because cells are preserved better in EDTA specimen. So here for sodium citrate especially we are using for coagulation studies itself for PT and APTT. So here why we are using sodium citrate instead of EDTA, because here especially sodium citrate is being used because of the factor 5 and factor 8, they are more stable in the citrus specimen rather than the EDTA specimen. So that's why we are collecting the coagulation parameters PT and APTT sample in sodium citrate. Now the next is heparin. Heparin is the only anticoagulant. Anti means against, coagulant is nothing but against coagulation. It will not get clotted. Okay, used to prepare samples for blood gas analysis. Blood gas analysis, that is arterial blood gas, pH, PO2, PCO2 and HCO3. These are our blood gases. These are arterial ABG, which we called our arterial blood gases. So heparin is the only anticoagulant which can be used to prepare the samples for blood gas analysis. Okay, now the last one is ESR. We have the ESR tubes also, right? ESR stands for erythrocytic sedimentation rate. In ESR tube you will be having 3.8% of trisodium citrate. Okay friends, so this is regarding your order of draw. Now you will be, I hope you will get cleared with why we are collecting in EDTA, why we are preferring anticoagulant like sodium citrate for PT and APTT, because for PT and APTT samples, the factors, the coagulation factors, factor 5 and factor 8, they are more stable in citrate specimen. I mean sodium citrates. That's why we are collecting coagulation parameters in sodium citrate. And for EDTA, against clotting. Okay, and for gray tube and yellow tube, for sugar samples you can use both gray tube and also yellow tube for the sugar parameters. And for heparin we are using for arterial blood gas, that is all the blood gases, arterial blood gases or venous blood gases or anything. Okay, next for erythrocytic sedimentation rate also we are using the Winthrop or blacktop vacuitainers. So these are the first we have learned the sequence order of blood draw. And we have, if anyone ask you regarding, if anyone cross question you that why we are collecting the sample in EDTA, why we are collecting the sample in sodium citrate, why we are collecting the sample in heparin, why we are collecting the sample in ESR. Okay, so these are the clarification you can give. Okay friends, so this is your basic knowledge regarding the order of draw. First you have learned order of draw. Second thing, why we are collecting specific sample in a specific container, and what are the additives and what are the additives helpful in. Okay, so now we are going to studying regarding the mixing of a sample. In mixing of a sample, nowadays we are having a large number of samples, so we can mix the sample in a sample rotators also. And sometimes for immediately when you if you cannot put the sample immediately in the roller, simply after extracting the blood from the patient, simply you can mix the sample with your hand in a inversion position. So here, as we have discussed regarding the different types of tubes, and the different types of tubes, they are having different types of mixing methods. Just like for culture tubes, you have to mix for 8 to 10 minutes. Okay, for sodium citrate 3 to 4 times, and for if you collect the sample in red plain tube, it should mix 5 times. Okay, and SST 5 times, heparin tube 8 to 10 times, EDTA 8 to 10 times, sodium fluoride also 8 to 10 times. Okay, inversion position. And friends, you should not shake the sample very rigorously, because sometimes it is RBCs, they may get hemolyzed. Okay, so slowly you have to mix the sample. So this is the order of draw, and this is the all kinds of additives which we are using, and why we are using, and how many times we are mixing the sample. So first thing you have to remember is order of draw. Okay friends, and the second thing is the additives, in which vacuitainer which additive we are using. For EDTA purple top we are using EDTA itself, purple top EDTA, dark green top heparin, light blue top, that is sodium citrate, gray color top sodium fluoride, SST is having a gel SST, that is gel separator, and plain we don't have any. In red top plain we don't have any kind of additive. So these are the different kinds of vacuitainers we are using in our lab for different kinds of purposes. So this is the small video on order of draw and kinds of and their additives. I hope you like it. And before starting this preparation, I want to share these things with you. So yeah, this is the video. Next video we will be starting the general microbiology topic, as we have begun our preparation series. Hope you like it. Please like, share and subscribe to my channel. Keep supporting me. Thank you guys.